So I have a new day job as a CEO of a new N of one nonprofit. I'm not going to talk about that today. Instead, I'll tell you about how Perlara is going to transition from 1.0 to 2.0, and it's through this, this story. So can everyone see my screen?
Yes.
Okay, I'm just going to go to full screen mode and I'll take it away. So I'll tell you today about individualized drug repurposing, going from bench to bedside in a year. And I want to hopefully leave the impression that this is something that's not wasn't a fluke occurrence, but something that can be repeated, not just for the class of diseases I'm going to tell you about, but I think more broadly for inborn errors of metabolism, which I think are relevant today. So the star of the story is a little girl named Maggie, and she's from Michigan. She has a congenital disorder of glycosylation called PMM2. It's been referred to once upon a time as CDG1A, PMM2 deficiency. PMM stands for phosphomonomutase, so it's a very core conserved enzyme, but usually goes by the name nowadays PMM2 CDG. So that's how I'll refer to it. So Maggie and her family represent a community that's probably 1,000 or more patients globally, and most of them are kids like her. And Maggie's family embarked on a cure odyssey, what we used to call a Pearl Quest at Perlara 1.0 3.5 years ago. And today, Maggie is the first PMM2 patient in the world taking a repurposed drug that we, and I emphasize we, as in her family and Perlara, discovered together. And I'm happy to report that the drug is working. Now, this is a picture of Maggie back in March before lockdown, the last conference, in-person conference I attended, which was for the CDG community. And she's been taking a drug called a Perlstein, and you'll learn more about this drug in a few moments. And I want to just show you a before and after video. And this is a video taken by her mom, Holly, and she's going through some sight word exercises. But you'll notice, obviously, her movement and her physical presence, and hopefully it'll be striking kind of the before and after. And so this is about 3 months before she started taking a Perlstein, and then the second video will be 3 months after a Perlstein. It's about a 2-minute video, so I'll play it now.
Good job. What letter is this? Got hair in your mouth? Get it out.
K.
K. Great job. What sound does a K make?
Tickle, tickle, tickle.
It doesn't say tickle, tickle. What sound does K make?
Who is that?
It's a kangaroo. What sound does K say? Ka, ka, ka. Good. Oh, this is. Say hi, everyone. Maggie is learning to read. So she has been practicing some sight words. There's 20 of them she's been practicing. And we've been using the snap words. And the snap words have a little bit of a symbol to help. So for example, you can see this word. All right. Okay, perfect. So here's the first word. Okay, Maggie, what word is this?
Me.
Me. That is right. You got it right. Good job. Okay, next word. What word is this?
Go.
Go. Awesome. You are so smart. Okay, what's this word? On. Great job. On. Okay, what's this word? Bye. Oh my gosh, you are so
So I hope it's pretty clear to the naked eye that she's obviously got more stability, more coordination, and as I can give you other examples. Her speech and cognition also seem to be improving. Um, and here's an example of a drawing she did pre-Perlstein, and she really wasn't actually capable of actually coloring. And now you can see a picture before. And Holly shared this on Twitter, and your very own Laura started a conversation about this, and asking some questions, whether this was improved fine motor control or cognitive gains or both. And I think the line that really struck me was Holly saying, I can have real conversations with her. So I think it's pretty clear the drug is having an effect. We have reported no side effects to this date, and she's been on the drug for over 6 months. So how did we get here? So this is, I think, the part that will interest everybody. I didn't want to bury the lead. I wanted to show the clinical data first, but now I want to tell you how we arrived at this place. And it really was courtesy of a trio of patient avatars, or another fancy way of saying disease models. And the disease models that we used were yeast, worms, and fibroblasts. And we think that's sort of this minimal set of models that could be very, very useful, especially in inborn errors of metabolism, where the underlying biology is faithfully conserved, even in a simple single-celled organism like yeast. And you also don't need to necessarily utilize complicated organoids or other tissue types that are human-derived, because again, this is a core enzymatic pathway that's operational in every cell of every body at all times of life. So we also took advantage of the fact that because this pathway of glycosylation is very well understood and well characterized, we could actually understand what's happening at a phenotypic level and connect it to the genotypic level, and try to actually even have a biochemical model that explains sort of comprehensively what's going on in the disease state. So I'm showing you here 3 examples of variants that are not real variants yet, but let's just imagine we've got this red, purple, and green flavored mutation. And in fact, we can map those onto the crystal structure of the PMM2 enzyme. And the PMM2 enzyme comes together the way sort of the two halves of imagine that my fist is one monomer, or one PMM2 molecule, and the other fist is another, and they actually come together in this fashion, where they actually have to form this interface, this so-called dimer interface, and that's how the enzyme properly functions. And we can map the location of these different mutations in DNA space to the protein space, to their location as amino acids in the protein. And not only can we do that, but we can also ascertain sort of what's going wrong with these mutations. So as I mentioned, there's different flavors of biochemical defect that manifest depending on what your genotype is, and that's not uncommon in this disease space as well. And so one flavor of mutation would be these dimerization defective mutations, where the dimers don't actually form stably and they fall apart, and so the enzyme doesn't function. You could have a mutation that's so-called folding defect, where the actual monomers themselves don't stably form and they fall apart, and so you don't even have an opportunity to form a dimer. And then you could have a case where each monomer folds up correctly, they're stable, forms a nice cozy dimer, but there's an Achilles heel where we mutated the so-called catalytic site, and the enzyme can no longer function. And we can model all of these variants in yeast. And the nice thing about yeast is we don't have to do anything more than just ask, how is the yeast growing? And when you implant these mutations into the yeast genome, they cause growth defects. And I'm sort of showing you here in arbitrary units, kind of relative growth defects next to each other. So a more severe mutation in a human would cause a more severe growth defect in a yeast. And we sort of established that relationship. And by doing so, we were able to take advantage of the beauty of yeast, which is that you could create many different genetic flavors of yeast with sort of different biochemical defects at the root of the disease, but they all show a growth defect. So you can use one common high throughput so-called phenotypic screening readout to be able to understand if you found drugs that could rescue the sick yeast. And then of course the workflow would be test the hits in yeast in a higher organism like worms, and then go straight to fibroblasts, because actually there is no viable mouse model for this disease. It's really tricky to model in rodents. And so we actually think we can skip right to the fibroblasts and essentially replace all those other organisms in between. And I'm not going to summarize all of this data. This is actually all published, but I'll just sort of summarize that we conducted a screen of about 2,600 compounds, including many approved drugs. And we found that there were 3 hits using those different, using a combination of about 6 different PMM2 yeast models. And one class of hit we found is this top row, represented by this compound that's actually the active ingredient in cinnamon. And this compound seemed to rescue mutants that had a dimerization defect preferentially over mutants that had unstable monomers. And we found actually a second class of drug that's actually the active component of black tea. And it had the opposite profile. It seemed to preferentially rescue mutants that had this folding instability over mutants that had a dimerization defect. And then we found this last drug, which actually is really a drug, so I'm using that word faithfully. This was an approved product called Suramin, which actually approved for African sleeping sickness back in the early 1900s. And it seemed to actually rescue sort of with no preference to the mutation. So it equally rescued the dimerization defective and the the protein folding mutant. However, this is a drug that's given for a very, it's not really appropriate for this disease area, and the other two compounds are natural products. And so we were sort of left with, well, we'd like to know a little bit more about what these drugs are doing, but we need a more informative model. And so we switched gears and created a worm dimerization defective allele because we had a hunch that that cinnamon compound represented an interesting mechanism of action. And here we're just sort of doing, and this is work that Sangeetha was first author on, so she can tell you more about this as well. But what we did here was just establish that we generated a bona fide PMM2 patient avatar, and this is just showing you the enzyme activity of PMM2 in these mutant worms in orange compared to the wild type in blue. And then we looked at a different congenital disorder of glycosylation called NGLY1 deficiency in gray, and it didn't have any change in PMM activity. So this gave us some confidence that we had generated a bona fide PMM2 worm model, the first of its kind. And we wanted to do an analogous screen that we had done with yeast, but now taking advantage of the fact that worms are more complex, they can develop. So you could add baby worms to a well of a 3 to 4 well plate, and then you could watch them develop over time. They could even produce offspring in the course of a week-long experiment. And so what we asked was, could we find molecules using the same collection we screened the yeast models? Could we find molecules, hopefully overlapping molecules, that rescued the growth and development defects in this worm dimerization defective mutant? And the worms themselves didn't really have a growth defect without any provocation. So we had to kind of make them a little upset by adding a compound called bortezomib, which is a proteasome inhibitor. And we had some other inklings as to why that would be a good idea. But in any case, I'm just sort of showing you in the upper panel a picture of a population of worms that are treated with bortezomib, but they don't really seem to be that affected. And then you can compare them to these PMM2 mutant worms at the same concentration, and they're pretty much wiped out by this treatment. And then the B panel just sort of shows you a dose response where we looked at across many, many different escalating doses, and we found a separation in the light gray versus the dark gray boxes where the PMM2 mutants were more sensitive. So we picked this regime of 13.6 micromolar bortezomib to ask, could we find any small molecules that could make these sick worms healthy and develop normally again? And so here I'm just sort of showing you examples of what sort of benchmarks are. So PMM2 worms alone, without any bortezomib, I said they sort of grow pretty robustly without provocation. But if you dampen their proteasome activity with bortezomib, you can see that you basically prevent these larvae from developing to adulthood, and so you don't see any progeny, and the larvae are sort of stunted and misshapen and not doing too great. And then this third well here from the left represents an example where you have a suppressor compound that restores growth and development basically back to normal. And just so we could show that the assay is working, we can even find so-called enhancers, which don't really have any therapeutic utility. They make the situation much worse. And this is just sort of zooming in so you could take a closer look at sort of what the phenotypes we were investigating. And using simple computer vision, you can just sort of identify all the pixels in this space that are occupied by a worm versus not. And you can also quantify the number of animals, their size distribution, and so forth. But you really didn't need that much work to be able to sort of see by the naked eye which compounds contain a suppressor or potential therapeutic agent that is making these PMM2 worms healthy again. And when we got the hit list, unlike in yeast where we only had 3 hits, here we had 20 different hits. And I've organized them a little bit by these shaded boxes. I won't go too much into the detail here. Suffice it to say that there weren't any direct overlapping hits between the 3 yeast molecules and these 20, but there was overlapping in terms of sort of family resemblance. These are all these so-called polyphenol compounds. And you might have heard of these compounds before because they turn up a lot in fruits and veggies and over-the-counter supplements. But we were sort of left with a position of, well, what are we supposed to advise as a family? You know, make a smoothie with these various components, or just buy these over-the-counter supplements and sort of run your own clinical trial. That didn't really seem like that was all that much different than what these families are doing already. So we had an insight where we realized, well, actually, the structural family resemblance of all these compounds really traces back to a substructure called p-coumaric acid, which is shown here in the middle. And that's a known aldose reductase inhibitor. And I'm just showing you on the left here a crystal structure of aldose reductase, and then showing you here just a number of related compounds, including curcumin compounds. So the compounds you might find ultimately in turmeric and other spices. So there definitely was a recurring theme here. But what was really interesting was there was another aldose reductase inhibitor, a synthetic version called the Perlstein, and that sort of struck our eye because it made us think, well, what could we do with an actual known drug? Because it turns out a Perlstein is an approved drug, not in the US, but in Japan. And it was actually approved for the treatment of diabetic neuropathy in adults, not patients. But when we sort of stared at the structures long enough, we could see that the cinnamon compound, the most exciting hit from the yeast screen, actually is really very similar to a Palrostat. And in orange, I sort of highlight this cinnamyl group, and then in blue, this carboxylic acid group. And you can sort of see here that the chemists at Ono, inadvertently or not, basically put in this spacer in between the cinnamyl and carboxylic acid groups of the natural product, and that's how they yielded this drug called the Palrostat, or what the trade name is Kindadak. And so we were able to show that side by side, a Palrostat and this CHCA, the cinnamon compound, could rescue or boost PMM2 activity in both worms and in patient fibroblasts, about 40 to 50%, at least looking at the genotype we were examining here in both systems. And when we examined some more fibroblasts, including in blue here represent Maggie's fibroblasts actually, we were able to show that there was a 4-fold increase in PMM2 activity. And when we average across these 3 different fibroblasts that all share this so-called catalytic null mutation, R141H, they all on average showed about a 2-fold increase. Now we subsequently went back and revised this assay a bit, and some of these readings here on the baseline are probably lower than they should be, but when we've done these experiments, you basically see about a 50 to 100% increase in PMF2 activity. So I wouldn't really trust the 400%, but we do definitely see a doubling in some instances, including in Maggie cells. So that really gave us some confidence that there was a direct activity on the enzyme that's deficient in this disease, and that we thought that it was worth testing this in a human because that percentage increase seemed like it should have therapeutic benefit. So then we went through a series of steps. First, getting a single patient IND or compassionate use to allow us to be able to treat a child with this drug. No child had ever been treated with this drug as far as we know, certainly no child with PMM2 deficiency. And then we did some other check boxes to get an orphan drug designation and a rare pediatric review, or a rare pediatric disease designation. I can go into that in the Q&A, but these are the kind of checks the boxes we checked on this path to getting this, the best 3 words you want to hear from FDA, which is study may proceed. Maggie's 6-month visit at Mayo was actually last week, but this is a picture of her first dose back in the end of January. I can report, as you saw with your own eyes in that video, that a Palrostat is working. We see improvements in coordination, cognition, speech, mobility. We even see examples of normalization of biomarkers, and there's a plan now to expand this trial. And I'll just sort of show you one of the biomarkers that's improved here. So in CDG, there's the protein in the blood called transferrin that gets hypoglycosylated. In other words, you don't have the appropriate numbers of sugars attached to this protein. And what you can see here is that at day 180, which was the 6-month visit, Maggie's showing green. And in other words, she's in the normal range for all of these transferrin readouts, including also this apolipoprotein, which is a different protein that people use. And that's, so you can see here, she has some red, some yellow. The first time she has all green was at the last visit. And here I'm just sort of plotting in the blue line that top value that is now at the normal range. So here's actually proof positive that we're improving glycosylation in Maggie on top of all the other changes that we're seeing with sort of the naked eye. And the remarkable part here is that sort of bearing the lead, but at the very bottom, interpretation, follow-up of a known patient with PMA2. In this sample, the transferrin and apolipoprotein were normal. That's the first time that word normal ever appeared in one of these reports. So this trial is happening under the stewardship of the incredible Dr. Ava Morava-Kozic, who's at Mayo Clinic. And here's a picture of Maggie and her parents at one of her birthday parties. I think this was 2 summers ago. She's going to turn 7, I think this month. And I don't have time to talk about this, maybe in the Q&A, but we're actually doing the same exact repurposing N of 1 with Ava and some other clinicians for NGLY1 deficiency. So again, this is where I think this is not going to be a fluke occurrence, and where I think, at least across the CDG space, and I think even more broadly in inborn errors of metabolism, this approach will work. So with that, it looks like I'm just on time, and opening up for questions.
This was amazing, Ethan. Thanks so much. It's really remarkable to see the amazing progress from the 2 videos. I do remember watching that and being kind of like blown away.
And sorry, here's just a little clip of Maggie on her bike just a couple of weeks ago, which really wasn't possible before. Anyway, so I'll pause here and take questions.
Yep, I'm gonna start adding the moderator. So for this session, which is our initial session, Sangeeta is a moderator and Heidi is a moderator. So we're just gonna have them start the Q&A. And folks, remember, you can use the Q&A box to post questions. I see we have quite a few questions there, and we can take turns and go from there.
Awesome. So happy to kick things off here, Ethan. So Heidi and I will be taking turns posting a question to you. And so maybe I can go first. Carol wants to know if you could explain how a drug can be repurposed. What kinds of disorders can it be used for?
I mean, everything's on the table. And I would say the opportunity is even more attractive and more accessible in cases where you don't have a tractable mouse model and people are beating their heads trying to make the perfect mouse model. This tells you that you don't even need a mouse model at all. And of course, you don't need to do any other rodent safety studies because this product was already approved. There was a question whether an adult drug would require some bridging safety studies when given to a child. But FDA basically, given kind of the sterling safety record of apalurestat, which is approved in Japan, China, India, it's been approved since '92. So lots and lots of patient hours that are, that resulted in safe usage of this drug, not to mention efficacious usage. So I would say the opportunity space is as broad as it can be, but I think it's especially within reach for folks who are struggling with that mouse model and who are struggling with, basically hitting a wall there and realizing you don't have to make the perfect human cell model as well. You can actually get by with fibroblasts.
So I have a quick question too. Do we know the mechanism by which this drug works?
Yeah, so I kind of glossed over it a little bit, but if I kind of go back to my slides here.
Sure.
So this is all unpublished work, but what I'm showing you here are 2 markers.
So I think you might need to share the screen again.
Oh,
sorry.
I'm sorry.
I had to quickly stop this.
So what I'm showing you here are normalization of 2 biomarkers, actually urine biomarkers, one of which is sort of reading out on a metabolite that should be going down if you actually are engaging aldose reductase with an inhibitor. So that's already one indication that it's acting as it says it does. We don't have to invoke some other pharmacology and some other target to explain this. It's sufficient to use, or it's sufficient to invoke aldose reductase inhibition to explain, I think, all the effects we're seeing. Although that in itself, aldose reductase inhibition in itself is a complicated thing, not to diminish that, but I think you can basically explain the effects we're seeing with apalurestat acting on label as an aldose reductase inhibitor, but those confirmatory mechanism experiments are still underway.
Great. Thank you. Heidi, go ahead.
I have a question from Aaron. What kind of tools were critical for this discovery path, and what kind of tools are critical to have available when considering studying a new disease?
So the tools available were basically just, so with yeast, the tools all exist off the shelf. So all you need to do is take a mutation and see if it's faithfully reproduced in a yeast. And then you don't even need to use CRISPR technology. You can use an older homologous recombination. They didn't call that gene editing back in the day, but that's a way to also edit the genome precisely. So that technology exists. You can whip up a bunch of personalized yeast avatars in a month. The worms will take some longer time. Sangeeta can kind of elaborate on that. We didn't make as many avatars in yeast in worms as we did in yeast, but that was using the CRISPR system and using a CRO, a contract research organization. So that is also something that's pretty commoditized at this point. And getting fibroblasts, like that's just up to you guys, right? Just go get a skin biopsy, and then you can establish a fibroblast line. So that's really all you need on the sort of disease model side. On the lab infrastructure side, you need to have some kind of an operation where you can perform these high throughput screens. And again, you don't have to have all of that purchased, infrastructure under one, under a proprietary roof. You could use contract groups, you could use collaborators, you can kind of put together a screening supply chain, if you will, that's academic based and even also CRO based. So yeah, the barriers to entry here are actually quite low.
Great.
Excellent. So one more question sort of allied to that is, if you had to apply learnings from this discovery path to say another disease, can you highlight maybe what was particularly difficult for other diseases or this one? What do you think was the secret sauce that helped you get this far?
I think the secret sauce was the fact that in yeast, this was such a well-behaved system. In other words, I'm not showing you all the control experiments we did to convince ourselves that the yeast being sick and slow growing actually was reflecting like what maybe a human cell, any one human cell. I mean, there's lots of tissues in Maggie, but like we think that the yeast we created are sort of in some ways representing the cell disease state that any one of her cells is going through, whether it's a neuron or a liver cell or whatnot. So I think that was really key to this, was making sure that the biology we were building on a really solid foundation. Because once that was solid, then the results in worm and fibroblasts, everything just sort of started to line up. So I would say the biggest learning here was really convince yourself that your disease models are faithfully reproducing the disease state, at least at a cellular level. And for the diseases we're talking about, like metabolic syndromes, these are sort of, each cell is kind of sick, and cells can't really cross correct each other. So you really have to address the problem in each and every cell. But again, if you're dealing with with really conserved biology, and you can convince yourself that a mutation behaves the same way in a yeast that it does in a human cell as it does in a worm, then that just increases the chances that you're not going to get stuck in a cul-de-sac that's a yeast-specific phenomenon or a worm, because we don't want to, as much as we might love yeast and worms, we're not in the business of curing yeast and worms. We're in the business of curing kids.
Thank you, Heidi.
And with the FDA, in your experience, how important or what weight do clinical biomarkers have versus patient outcomes?
So we didn't really probe that space because, as I said, we didn't really know what to expect. We kind of, Maggie's N of 1 has been like kind of this prototyping experiment, to tell us what would a larger trial sort of look like. So yeah, I mean, we kind of discovered along the way that there are these urine biomarkers that are not only easy to kind of get, especially compared to blood, but are also super informative about the way the drug is working and also about the cellular disease state that we're fixing. So we didn't have all that information when we started. All we had was, hey, this drug has a long safety record in Asia in adults, and we want to try it in this disease, and here's our rationale. This molecule boosts the enzyme activity that is deficient in these patients, and that seemed to be good enough. Now when we go back to them with an expanded trial and try to open up a proper IND, we can say, look what we learned. Maggie was a sentinel, so she'll always be 6 months or a year ahead of the next set of dose patients. And now using her experience allows us to understand, oh, there's this urine biomarker we can use. Oh, there's this fibroblast phenotype that copies the urine biomarker. And so there's a way now to even rationally select and enroll patients to maximize their chance of actually getting benefit from this drug. So none of that, I wish we had premeditated all that, but we didn't. But I think the flexibility of compassionate use in single IND, if you don't abuse it, is that you can basically use that first patient to kind of de-risk and prototype the larger clinical trial design.
So we have one question from Forbes Porter. How conserved is creatine metabolism in yeast?
Good question. So I knew I should have looked this up right beforehand. So I did not. I'd done it a while ago. So, I mean, it's very, very simple to ascertain the level of conservation, and very easy to ascertain whether the growth defects you see in yeast are coupled to the biochemical defect that's taking place in human cells. So I wish I had done that homework and can give you that answer, but it's, with a few searches and a few clicks, you'll be able to make the case yourself or not.
So as a follow-up to that, are you planning to look at a repurposing drug for CTD?
Well, so I don't have a lab. Perlara is a virtual company right now, and so I am sort of in talks with an academic medical center site to try to look at least at a mito disease and another glycosylation disease using this, not with worms, but using a kind of yeast to fibroblast workflow. And that's all I can sort of say in terms of what I can personally be involved in. But on paper, I think there's no reason why you couldn't kind of reproduce this story.
And I want to make a quick comment on like our research philosophy on the ACD and how we feel about this, right? So I have my 5.5-year-old is actually upstairs, right? Like I have a son with CTD, and I think my biggest goal as being part of the ACD is to make sure that we have many eggs and many baskets, right? So we have many avenues for research, and I'm a scientist in something completely different in my daily life, right? But I know that not everything pans out, right? And the whole I see our role as a patient org to go through a lot of, like fund a lot of these like failures, right? So that eventually we can get to an actual success. And drug repurposing is a great, it has a gap that we can perhaps support or figure out what could be done in that space, right? So I think we're very keen on plugging a lot of these gaps, and Sangeeta is going to talk more about a lot of the conversations we've had for the past couple of months of, what kind of tools can we support that and partnerships we can have with academics and industry to move multiple buckets and multiple baskets at the same time.
Yep.
And
So
I think sorry, go ahead,
we are
Sangeeta.
almost at time, but we have 2 more questions in the Q&A, and I think we can just take time to address those. So Heidi.
I have one that asks, is it possible this drug can work for mutations that are different than Maggie's?
Yes, and so far the data we have suggests that it's true. It's just that the magnitude of the effect seems to vary from genotype to genotype, from patient to patient, and we don't have a predictive rule set yet. But definitely, absolutely, we think this is going to be something that would be kind of broadly used, but some mutations, again, might respond better than others.
Excellent. And I'll take what looks like the last one over here, which is, given that this was a repurposed molecule, can you elaborate what kind of safety data you needed to start an N of 1 clinical trial?
Yeah, this much, 0. As I said, we thought that we would be encouraged to do a bridging safety study for children, but I can show you that letter. The FDA said you may proceed.
Great.
That's wonderful.
Thank you.
Well, thanks so much, Ethan, and thank you, Sangeeta and Heidi. This was. We're gonna take a quick break, a stretch out break. Zoom meetings are hard, so please go and stretch. And we're gonna play some more videos in the background. So come back in 10 minutes, and we're gonna have a new session. And
thank you again for your audience and for your time.
Thank you
so much.
Thanks so much, Ethan.
My pleasure.
We knew early on that something wasn't right with Reed.
I remember staying up late at night, and I did everything I could to try to figure it out.
The cerebral creatine deficiency syndrome are a collection of genetic conditions in which there is not a sufficient amount of creatine in the brain. As a result, the brain cannot function in the proper way. There are three known causes of cerebral creatine deficiency. Two are defects in the synthesis of creatine. The third condition is a defect in the transporter of creatine, the one that carries creatine inside the brain.
What they all have in common is that since creatine helps us to get energy to the right place at the right time in a cell, all of those patients suffer from a problem that is mainly affecting the functioning of the brain.
Probably about 18 months when he began developing seizures. We first noticed it too, but again, looking back, I think those seizures were happening earlier than we anticipated.
He had significant GI issues. He spit up a lot as a child.
Projectile vomiting, way beyond any normal reflux.
He was always under the growth chart as far as weight and height were concerned.
Sometimes there were trouble with initial feeding, such as latching right after birth when trying to breastfeed, sometimes a weak suck.
Typically, it's in the first 2 years that families recognize that the kids are not achieving all of the developmental skills as expected.
He wasn't crawling, he wasn't walking, he wasn't even attempting any of those.
These children usually are globally delayed.
Delayed language is often one of the things that parents and providers notice first as a problem.
She really didn't have any full words by the time she was 5 years old. That combined with absence seizures really started to kind of give us a heads up that something else might be wrong.
A patient has mild developmental delay and problems feeding. That is when you should start questioning if there are not other things going on that are actually uniting it.
One of the challenges to getting a diagnostic is this notion that there wasn't anything on fire. It's like being a detective and you're trying to latch on to what's that cue. And for creatine deficiency, it's very hard to find that. You don't have that big revealing sign.
As parents, we were desperate to find answers. We put him through so many tests and procedures. And at the end of the day, what diagnosed Reed was a simple urine test.